Good day everyone. This is the part 2 of our presentation or the second part of our report. And I hope you will continue listening as we will be learning a lot of things together. Serology is the science that studies serum and immune responses that are evident in serum. Microorganisms are antigenic, that is microorganism that enters animal's body, stimulate it to form antibodies. Antibodies are proteins that circulate in the blood and combine in a highly specific way with the bacteria that cause their production. Serologic tests are blood tests that look for antibodies in the blood. They can involve a number of laboratory techniques, and this is associated with various types of serologic tests which are used to diagnose various disease conditions. Next slide please. Antiserum, or the plural is antisera, solutions of such antibodies used in the identification of many medical important microorganisms are commercially available. Slide agglutination test samples of an unknown bacterium are placed in a drop of saline on each several slides. The following figure shows a slide agglutination test. A. First is in a positive test. The grainy appearance is due to the clumping or agglutination of the bacteria. Letter B, or the second, in a negative test, the bacteria are still evenly distributed in the saline or antiserum. A person needs serologic tests because it helps to know about immune system and why a person gets sick. Antigens are substances that provoke a response from the immune system. They're usually too small to see with the naked eye. So what will happen during serologic test? During serologic test, a blood sample is all that the laboratory needs to conduct testing. Serologic testing can diagnose multiple illnesses, including the following. First is brucellosis, which is caused by bacteria, amoebiasis, which is caused by parasite, measles caused by virus, and rubella, which is caused by a virus, and also HIV, syphilis, and fungal infections. Serological testing can differentiate not only among microbial species, but also among strains within the species. Strains with different antigens are called serotypes, serovars, or viovars. And Rebecca Lansfield was able to classify streptococcal serotypes by studying serological reactions. She found that the different antigens in the cell walls of various serotypes of streptococci stimulate the formation of different antibodies. Next slide, please. A test called enzyme-linked immunosorbent assay, or ELISA, is widely used because it is fast and can be read by a computer scanner. This type of test commonly used to measure antibodies, antigens, proteins, and glycoproteins in biological samples. This includes diagnosis of HIV, or human immunodeficiency virus infection, pregnancy tests, and measurement of soluble receptors in serum. Next slide, please. Another serological test is Western blot. The Western blot proteins separated by electrophoresis can be detected by their reactions with antibodies. Another serological test, Western blotting, is also used to identify antibodies in a patient. This type of testing usually used to confirm a positive HIV diagnosis. During the test, a small sample of blood is taken, and it is used to detect HIV antibodies, and not the HIV virus itself. The Western blot is a test that separates the blood proteins and detects the specific proteins called HIV antibodies that indicate an HIV infection. So, the Western blot is used to confirm a positive ELISA, or the enzyme-linked immunosorbent assay. The combined test with the ELISA are 99.9% accurate. Next is page typing. Like serological testing, page typing looks for similarities among bacteria. Both techniques are useful in tracing the original course of a disease outbreak. Page typing is a test for determining to which a page a bacterium is susceptible. Page typing is most often used for the two common Salmonella types, the S. enteritidis and the S. typhimurium. But page typing system also exists for some other Salmonella serotypes and a few other bacteria. This type or method provides a rapid, accurate, and cheap method of investigating Salmonella strains for epidemiological use. Next slide, please. The figure shows the page typing of a strain of Salmonella enterica. The tested strain was grown over the entire plate, the entire plate. Next slide, please. Fatty acid profiles are currently most frequently determined by capillary gas chromatography of metal esters, which are prepared by a variety of methods. Next slide, please. Bacteria synthesize a wide variety of fatty acids. These fatty acids are constant for a particular species. Fatty acid profiles called FAME or fatty acid metal ester are widely used in clinical and public health laboratories. FAME or fatty acid metal ester are type of fatty acid ester that are derived by transesterification of fats with methanol. So, and again, the fatty acid profiles are currently most frequently determined by capillary gas chromatography of metal esters, which are prepared by a variety of methods.
Hi, I am Myla Tracy Cantoja, and today I am going to discuss three topics which are flow cytometry, DNA sequencing, and DNA fingerprinting. We will first talk about the flow cytometry. So this can be used to identify bacteria in a sample without culturing the bacteria itself. So the simplest method detects the presence of bacteria by detecting the difference in electrical conductivity between cells and the surrounding medium. As we can see on the figure, in a flow cytometer, a moving fluid containing bacteria is forced through a small opening. So, if the fluid passing through the opening is illuminated by a laser, the scattering of light provides information about the cell size, shape, density, and surface, which is analyzed by a computer, which can be shown on the screen. So this is an example of the computer which analyzes the data from the microorganism. So from the computer, from their data, so that is the example. So now we are going to proceed to the fluorescence can be used to detect naturally fluorescent cells such as Pseudomonas or cells tagged with fluorescent dyes. So a proposed test that uses flow cytometry to detect listeria in milk could save time because the bacteria wouldn't need to be cultured for identification. Right? The antibodies against listeria can be labeled with a fluorescent dye and added to the milk to be tested. So the milk is passed through the flow cytometer, which records the fluorescence of the antibody labeled cells. And that's it for the flow cytometry. And now we are going to proceed to the second one, which is the DNA sequencing. So the taxonomies can use an organism's DNA-based composition to draw a conclusion about their relatedness. This base composition is usually expressed as the percentage of guanine plus cytosine or the G plus C. So the base composition of a single species is theoretically a fixed property. Thus, a comparison of the G plus C content in different species can reveal the degree of species relatedness. So each guanine in DNA has a complementary cytosine. We all know that. Similarly, each adenine in the DNA has a complementary what? Thymine. Very good. So this is an example of a DNA helix in which we can see how they are being paired from each guanine in DNA has a complementary cytosine and an adenine paired to thymine. During the last decade, comparison of DNA sequences has led to great strides in reclassifying known species and identifying new species. So we have here a genetic sequences of hundreds of organisms are compiled in databases that can be used online through the NCBI Genome Database. So this is what their website look like. They have a all the information stored there. You can visit their website if you have any time or if you're curious. Okay? So now we're going to proceed. We can see on the screen the new Lyme disease pathogen called Borrelia mayuni, which was discovered by the researchers in 2016 with the use of the DNA sequencing. We are now going to proceed with the DNA fingerprinting after we discuss about flow cytometry and the DNA sequencing. We are going to discuss about this like determining the entire sequence of bases in an organism's DNA is impractical for laboratory identification because it requires a great amount of time, right? We all know that. However, the use of restriction enzymes enables researchers to compare the base sequences of different organisms. Restriction enzymes cut a molecule of DNA wherever a specific base sequence occurs, so producing restriction fragments. So this is what a restriction enzyme looks like, from restriction digestion of vector to restriction digestion of gene of interest to ligation to transformation and through culturing. So for example of that is the enzyme E. coli cuts DNA at the arrows in the sequence GAATC and the CTTAAG. In this technique, the DNA from two microorganisms is treated with the same restriction enzyme, and the restriction fragments, or the RFLPs, produced are separated by electrophoresis, producing a DNA fingerprint which can be shown on the image. Right? That is what it looks like. The image shows that comparing the number and sizes of restriction fragments that are produced from different organisms provides information about their genetic similarities and differences. The more similar the patterns or DNA fingerprints, the more closely related the organisms are expected to be. Understood? So DNA fingerprinting is used to determine the source of hospital-acquired infections. In one hospital, patients undergoing a coronary bypass surgery develop infections caused by the Gordonia bronchialis, which is shown on the picture. So that is the source of infection in the hospital. So the DNA fingerprints of the patient's bacteria and the bacteria of one nurse were identical. So, the hospital was thus able to break the infection's chain of transmission by encouraging the nurse to use aseptic technique. This has led to interest in finding a few genes that are present in all species and that provide a large variation between species. Primers for these genes would be used for PCR to produce a DNA barcode for each species. This was first proposed in 2003 for eukaryotic species, but the necessary 69 genes for bacterial identification have not been found yet. And that ends my report for the flow cytometry, DNA fingerprinting, and the DNA sequencing. And now let us proceed with the next reporter.
Hi everyone, I'm Danica NW and I'll be reporting the nucleic acid hybridization. Nucleic acid hybridization is the DNA chip. A new exciting technology is the DNA chip or microarray which can quickly detect a pathogen in a host or the environment by identifying a gene that is unique to the pathogen. See in the figure. A DNA chip can be manufactured to contain hundreds of thousands of synthetic single-stranded DNA sequences. Assume that each DNA sequence was unique to a different gene. Next, if a double-stranded molecule of DNA is subjected to heat, the complementary strand will separate as the hydrogen bonds between the bases break. If the single strand are then cooled slowly, they will reunite to form a double-stranded molecule, identical to the original double strand. And when this technique is applied to separate the DNA strands from two different organisms, it is possible to determine the extent of similarity between the base sequence of the two organisms. See in the figure, DNA, this is the DNA hybridization. The greater the amount of pairing between DNA strands from different organisms, the more closely the organisms are related. Next, nucleic acid hybridization can be used to identify a known microorganism by southern blotting. Rapid identification methods using DNA probes are being developed. See in the figure, a DNA probe used to identify bacteria. Southern blotting is used to detect specific DNA. This modification of the southern blot is used to detect salmonella. Next, the DNA chip is composed of DNA probes. A sample containing DNA from a known organism is labeled with a fluorescent dye and added to the chip. Hybridization between the probe, DNA, and DNA in the sample is detected by fluorescence. See in the figure, it is a DNA chip, and this DNA chip contains probe for resistance genes. It is used to detect antibiotic-resistant bacteria in samples collected from animals on a farm or in slaughtering facilities. Next, ribotyping and ribosomal RNA sequencing. Ribotyping is currently being used to determine the phylogenetic relationships among organisms. There are several advantages to using RNA. First, all cells contain ribosomes. Nucleic acid hybridization. See in the figure, fish are fluorescent in situ hybridization. A DNA or RNA probe attached to fluorescent dyes is used to identify chromosomes. Bacteria seen with phase contrast microscopy, A, see in the figure, are identified with a fluorescent label probe that hybridizes with a specific sequence of DNA in Staphylococcus aureus, B. Fluorescent in situ hybridization, or fish, fluorescent dye-labeled RNA or DNA probe are used in stained microorganism in place or in situ. Next is putting classification methods together. Morphological characteristics, differential staining, and biochemical testing were the only identification tools available just a few years ago. Technological advancements are making it possible to use nucleic acid analysis techniques once reserved for classification or routine identification. Dichotomous key are widely used for identification. In the dichotomous key, identification is based on successive questions, and each question has two possible answers. Dichotomous means cut in two. Cladograms. Cladograms are maps that show the voluntary relationships among organisms. Clado means branch. See in the figure, this is building a cladogram. Each branch point on cladogram is defined by a feature shared by various species on the branch. Historically, cladograms for vertebrates were made using fossils evidence. Our RNA sequence are now being used to confirm fossil-based assumptions. That's all. Thank you.